This video for my general biology students at RCBC is going to focus on gene regulation. It's a broad overview. There are other videos that give some more details. We also call this control of gene expression. If we take a look at this bacterial cell here, you can see basically what is the central dogma. DNA in the form of a gene is going to be used to create mRNA, which is going to be used to create proteins. In bacterial cells, the primary form of regulation happens at transcription. They can do a little at translation and afterwards with the protein, but they are very efficient and they're not going to even bother to transcribe a gene if they don't need that protein. These bacterial cells have molecules such as repressors. Repressors bind to the gene or a part of the gene or a place near the gene, I should say, and they prevent RNA polymerase from getting in there to the promoter and getting to the gene. And so repressors do not allow transcription to occur. There are also effector molecules that play a role. And an example of one of these effector molecules is lactose. So when lactose, as you saw in the other video, sits on the repressor changes shape. It can no longer get to that part of the DNA that it sat on, and therefore RNA polymerase can get to the DNA and the gene, and you get some mRNA that's made. Remember this crazy Lac Operon? This place right here is where the repressor sits. So it's not within a gene, it's an operator region, but it prevents RNA polymerase from getting to the genes. Lactose is a prime example of one of these small effector molecules. E. coli does not make the proteins to digest lactose if lactose is not present. And that is kind of foreign because it's not the way our bodies work for the most part. There is something that I did not discuss in the lactose or the Lac Operon videos, and I wanted to mention it here. Even when lactose is present in high levels, E. coli doesn't always turn on that Lac Operon. If glucose is present, it will keep the Lac Operon turned off. Now, I'm not going to get into why that is. It involves this capside and cyclic AMP, but just think of it this way. If lactose is a disaccharide, half of which is glucose, why would I bother to waste the energy of making enzymes to break down that disaccharide to get to the glucose if there's plenty of plain old glucose available already? So when glucose levels are high, we do not turn on the Lac Operon, whether lactose is present or not. We keep it shut off. The last thing that I want to say about control of gene expression in bacterial cells is about quorum sensing. Remember, we discussed this in a previous lecture. This is where molecules are sent out by bacterial cells, and they wait to receive similar molecules back from other bacterial cells. They are waiting to find out whether enough of them are present to bother turning on certain genes. For instance, fluorescence genes in Vibrio fischeri or toxin genes in Vibrio cholerae. They know they can't really have an impact if there's only a few of them. So they create these molecules sort of like a smoke signal, sending them out to other bacterial cells and waiting to see the smoke and feel these molecules from their neighboring bacterial cells. And then they all say, okay, it's time to turn on gene expression, time to make these proteins. Moving on to eukaryotes. Eukaryotes have a few extra steps, as I mentioned previously. They control for sure at the transcriptional level, but they also can control at this RNA modification. And in large part, that does not exist in bacterial cells. They also do more at the post-translational area. For instance, sometimes you have a protein that needs a sugar added to it, and that's called glycosylation. Glycosylation doesn't really happen in prokaryotic cells, and so that's another unique part of controlling functional protein in eukaryotes that you would not see in prokaryotes. So let's take a look at some specifics here. We mentioned in previous lectures that we have general transcription factors. Those general transcription factors in yellow here find the promoter. Then remember the TATA box is part of the promoter. Then they call in the RNA polymerase. But we're also going to see that we have some other molecules as well. We have these activators, and activators, which we can see right here, they bind to enhancers. I often talk about enhancers, but I call them switches. And the enhancer region doesn't have to be right next to the promoter and the gene. It can be 1,000 base pairs away. And so you can see that up here as well. And what this shows us is that even though it's 1,000 base pairs away, when an activator sits on that switch, on that enhancer, it can interact with the general transcription factors through the use of this mediator in green. And so you can see a bend in the DNA where the activator, the mediator, and the general transcription factors come together in close proximity. There is a lot of detail I'm leaving out about these general transcription factors, but you don't need to know that. Whoops. Okay. Another thing that happens is in eukaryotes is that we have alternate splicing. And of course, that cannot happen in prokaryotes because prokaryotes don't have introns. They don't splice. So, but what I'm showing you here is that I can use one gene and I can create two different proteins or two different pieces of mRNA, which will make two different proteins based on which of the exons I keep and which ones I splice out. So when I'm talking about splicing, I previously was talking about splicing out the introns, but in this case, we're also splicing out some of the exons. I hope this helps to briefly show the differences between how you can control bacterial gene expression versus eukaryote gene expression. Thank you very much.